Hello, welcome in this video lecture. In this lecture, we are going to discuss a special term that is enzyme specificity, different classes of enzyme according to their functionality, and third sub point that is the factors affecting the catalytic activities of enzymes. Now, in previous point, we have discussed how the rate of substrate increased in presence of enzyme as a catalyst by its reaction progress or graphical representation and mechanical diagram of substrate, enzyme, active site, what is substrate, like this. So enzyme specificity is nothing but the enzymes are specific, particular whenever they increase or catalyze the substrate reaction rate. Their specificity is due to both the selective geometry of their active sites and their ability to lower activation energy for particular substrate. So, enzyme specificity is simply defined as the capability of enzymes according to their selective geometry of active site and ability to lower activation energy of particular substrate. They are having tendency to react with particular substrate, and this is called as specificity. Very simple definition. For example, an enzyme with absolute specificity is urease. Urease. Urease is the one of the enzyme which is used by some of the plants, fungi, bacteria to catalyze the reaction in which urea, only urea, is converted to ammonium and bicarbonate salt. See, the enzyme which converts urea into ammonium and bicarbonate salt is named as a urease. Urea, urease. So, specificity of urease enzyme is that by the plants or fungi or bacteria, whenever they get urea as a substrate, this urea is converted into ammonium or bicarbonate salts. So, specificity is having the tendency to catalyze specific substrate reaction. This is one. Second thing, some of the enzymes are less specific. They are having specificity very low. Will catalyze reaction for a particular family of substrate, and this is called as substrate specificity. Only specific substrate. This is called as substrate specificity. And for example, substrate or relative specificity. For example, suppose enzymes with relative specificity include protease. Protease is the one of the enzyme which catalyze the hydrolysis of various proteins. Hydrolysis of various proteins is carried out with the help of enzyme called as known as protease. Protease, protease, and it is specific for hydrolysis of protein. So this is the specificity known as relative specificity, right? Now second type of specificity is the stereo specificity. See another example, stereo specificity. Now what is stereo specificity? those enzymes which catalyze the reactions of various substrates and give the product of particular stereoisomer. These are called as stereospecific enzymes. Stereospecific enzyme. According to substrate stereochemistry, this substrate combines with specific enzyme and they gives formation of specific particular stereoisomer only. If they are cis, they give cis. If they are trans, substrate are trans, they give trans stereoisomer of product. So such kinds of enzymes are called as stereospecific enzymes. Specificity two types. One is the relative specificity and another one is the stereospecificity. The example of stereospecificity is, now see the example, one of the large molecule is shown here. Whenever one of the large molecule is shown here, whenever enzyme with specificity is, stereospecificity is steroid coenzyme A9 desaturates. This is the name of enzyme. Remember that. Steroid coenzyme A9 desaturates is an enzyme that is involved in fatty acid metabolism and is present in every cell in the body. Now, how is catalyze the reaction? See the reaction now. This is the large molecule which is converted into catalysis of this disaturates into its cis isomer, and this specificity is the stereospecificity. Stereospecificity is nothing but a specific stereoisomer of substrate is converted into specific stereoisomer of product. For example, cis gives cis, not trans, trans gives trans, not cis. So it is called as stereospecificity. So this is about the overall about enzyme and its specificity. Now we are moving towards the classification of enzymes according to their functional group or which kind of functions they are carried out. Now, almost all enzymes named use the suffix, that is, remember which suffix is used, ASES. ASES. How it is? Isomerization reaction is carried out in presence of isomerase. What is the suffix? ASES. And this ASES indicates that this is enzyme and functionality increases. Suppose isomerization is carried out, name of the enzyme or class of the enzyme is isomerase. So almost all enzymes names use the ASES as a suffix, right? So enzymes are named and categorized based on their functions and the use of their they catalyze or substrate they catalyze. Now see, a classification is shown in the chart likewise. First column shows enzyme class. First class of enzyme is isomerase. Isomerase, first class. Which kind of reaction it involved? Catalyze rearrangement reaction. The reactant and the product contain same atoms, only the bonding pattern is changed. Means, for example, cis trans isomerases convert to cis to trans or vice versa. Cis to trans or trans to cis. So Isomerase enzyme converts or carry out isomerization reaction, and cis isomer is converted into trans isomer is converted into cis. Such kind of enzymes are known as isomerase. First class. Second, ligases. 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 Now, what is its reaction? It is used energy from ATP, adenine triphosphate, to form chemical bonds between substrate. Ligases type of enzymes forms bond with substrate, and DNA ligase forms bond between two DNA fragments. Such kind of class of enzymes are called as ligases. Third is hydrolysis. Hydrolysis means what? Hydrolysis. These are used for hydrolysis reaction. Hydrolysis catalyze hydrolysis reactions. And example of hydrolysis are lipases, hydrolyze ester bond into lipid, proteases, hydrolyze peptides bond, phosphatase, hydrolyze phosphoester bonds, nucleases hydrolyzes bond in DNA and RNA, and carbohydrates hydrolyzes glucosidic bonds. So these are the third class of enzymes known as hydrolyzers. Then next come oxidoreductases. Oxidoreductases. No doubt, according to its name, this enzyme catalyzes oxidation reduction reaction. And examples are oxidases, oxidize a substrate, reductases, reduces the substrate, dehydrogenase remove two hydrogens from neighboring carbon and form a double bond. So these are the oxidoreductase. And last one, very important one, that is the transferase. Now, what is the transferase? Catalyze the transfer of a group of atom. It catalyzes transfer of group of atom. Kinases and transaminase. See the two examples. Kinases and transaminase are the transferases. What is the role of kinases? Transfer a phosphoryl group, PO3 minus phosphoryl group, and transaminase transfer the amino group. So this is the functional groups are transferred. Now these are the five important classes of enzymes: isomerase, ligases, hydrolysis, oxidase, and transferase. Their utility of reactions and examples are we have studied. Now this point we have to learn, that is the classification of that particular compounds or enzymes. Now next we are moving towards another point, that is the very important point: factors affecting the rate of reactions of enzyme catalysis. Now we have seen enzymes are catalysts. They are having tendency to increase the rate of reactions. But again, there is some factor which decide whether the reaction rate is increased, decreased, or it remains moderate. So there are some of the factors that affect the rate of enzymatically catalyzed reaction. And first important factor is the temperature, effect of temperature. It's very important one. See, I am taking a general example. In the winter season, we the human being are very energetic. Winter season, energetic. We are doing so many exercise and every physical activity. But in summer season, our physical activity and exercise activity are having some moderate. So, in the same manner, effect of temperature on rate of enzymatically catalyzed reaction is very important one, and it is shown in the graph. See the graph, reaction rate versus temperature. A typically graph of rate of enzymatically catalyzed reaction versus temperature in degree Celsius is shown in the slide. The temperature at which rate of reaction is highest is called as enzymes optimum temperature, right? See the graph. In the graph, optimum temperature is shown. It is near about 37, right? 38, 37. So, optimum temperature is the temperature of enzyme catalyst reaction at which the rate of reaction is highest. And this temperature is known as optimum temperature. So, according to the effect of temperature, at optimum temperature, most of the specific enzyme catalyzed reaction, specific, the rate of reaction is higher. And the reason is that initially, see the temperature 0 degree, then 10 degree, 20, 30. The nature of graph is increasing. But at optimum temperature, it becomes highest and later on it goes on decreasing. Rate of reaction goes on decreasing, and it happens above optimum temperature, that is 40, 50, 60. There occurs denature of enzymes. That is, enzymes are get denatured at higher temperature. So, up to optimum temperature or at the optimum temperature, the rate of reaction goes on increasing, but later on it is get decreased, and this decrease in observe that at very highest temperature the denaturization of enzyme takes place, enzyme becomes deactive, and hence an enzyme's optimum temperature is usually very close to the normal temperature of the organism in which it exists, and hence in the diagram those temperature is shown, it is near about 37. So in the first factor, effect of temperature, rate of reaction increases up to certain temperature, then it again decreases specific temperature at which rate is highest, known as optimum temperature. So this is very important. First, that is optimum temperature. Now the second factor is optimum pH, right? Optimum pH. See the graph. See the graph. Similar to optimum temperature, the effect of pH, that is reaction rate versus pH, shows that the optimum pH is the pH of enzyme catalyzed reaction at which rate of catalysis of that substrate reaction is very highest, and such pH is called as optimum pH. Again, optimum pH is usually close to normal pH of the region of organism in which the enzyme exists. Now see the graph. In the graph, vertical axis shows reaction rate, horizontal axis shows pH of enzyme catalyzed reaction solution, starting from 4.0 pH to 10.0. At 7.4 pH, the rate of enzyme catalyzed reaction is highest, and later on it goes on decreasing, still the pH of solution increases. So pH increases or decreases from the optimum pH, the reaction rate is lower than rate at the optimum pH, and it happens again due to denaturization of enzymes. So this is the second important factor, that is the effect of pH, the rate of reaction, and here you have to study the graphical representation importantly. Now the graph which is shown in the diagram is the digestive enzyme of pepsin. Pepsin is the digestive enzyme used to digest the food taken by human body, and digestion optimum pH is 7.4 for digestive enzyme, that is pepsin. Pepsin is having optimum pH 7.4, which functions in our stomach has an optimum pH of 2. Normal pH is 2 in the stomach. pH ranges normal range 1 to 3. It is not surprising that digestive enzyme called as pepsin, which is having function in the stomach and has an optimum pH of 2, but overall reaction of our body is the 7.4. So this is the second factor that affect the particularly various kinds of important one. So these are the two important factors, and third important factor is control of enzymatic reaction by inhibitors. Now see the definition of inhibitors. All life forms control of enzymatic reactions. There are important substance which inhibits the enzyme catalyst reaction. One is the enzyme inhibition. Now what is the inhibition? When particular molecule forms a covalent or non-covalent bond with enzyme, it can result in decrease in enzyme's activity. That is, in enzyme inhibitors are nothing but the substance which decrease the activity of enzyme catalyzed reactions. Such type of substances are called as enzyme inhibitors, and that control the enzymatic reaction. This is the third factor, and there are two types of inhibitors. One is the irreversible inhibition, and another one is the particularly enzyme inhibitors irreversible nature. Whenever inhibition takes place in irreversible manner, then such kind of inhibition is called as irreversible inhibition. Okay, so inhibition processes also control the rate of reaction. So there are three important factors that affect the rate of reaction: optimum pH, optimum temperature, that is effect of temperature or effect of pH, and third important factor is the control of reaction by inhibitors. Enzyme inhibition means decrease in rate of enzyme catalyzed reaction. So this is overall about the three important points of enzyme catalyzed reactions. Okay, thank you.